Hello, everyone, and welcome to today's live broadcast, The Role of SIRT1 LXR Alpha Beta Signaling in Diabetic Retinopathy, presented by Dr. Sandra Hammer, Research Fellow, Michigan State University. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Thermo Fisher Gibco. Gibco Cell Culture Heroes is a program putting PhD and postdoc researchers around the world in the spotlight. For more information on our sponsor, please visit thermofisher.com/cell culture heroes. Now let's get started. I would like to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. You can also see at the top of the page multiple tabs. Please click through our related applications, upcoming and on-demand webinars, as well as links to the Cell Culture Heroes webpage. You may be our next Cell Culture Hero. Also, please notice that you can share this webinar on your personal social media. Just click on the social sharing tab to let your friends and colleagues know about today's live event. If you have trouble seeing or hearing the presentation, click on the support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of your presentation window and follow the process to obtain your credits. I would now like to introduce today's presenter, Dr. Sandra Hammer, Research Fellow, Michigan State University. Dr. Hammer is a postdoctoral fellow in the Busik Laboratory at Michigan State University. Prior to being at MSU, she attended Vanderbilt University, where she obtained a PhD in Cell and Developmental Biology under the mentorship of John Penn. Prior to this, she received her BS degree from the University of Miami. For a complete biography on Dr. Hammer, please visit the Biography tab at the top of your screen. Dr. Hammer, you may now begin your presentation.
Great, so thanks so much for that introduction, Christy. Hi everyone, my name is Sandra Hammer. I am a postdoc in the Busik Lab at Michigan State University. So before diving into my research, I just wanted to thank the Gibco and the LabRoots teams for this awesome opportunity today to share my work on this platform. So today I'm going to be talking about the research that I'm doing in the Busik Lab, and it really centers on lipid dysregulation, specifically looking at cholesterol metabolism in diabetic retinopathy, which we will get into in a little bit. Although my current work in the Busik Lab focuses on dyslipidemia and cholesterol, trying to understand these early driving mechanisms of diabetic retinopathy progression have really been of great interest to me since I started graduate school in 2011. And in 2015, I got my PhD in Cell and Developmental Biology from Vanderbilt. And I was looking at the effect of high glucose in retinal pericyte apoptosis, and this is interesting to me because retinal pericyte apoptosis is one of the first histopathological events that happens in the disease, so it's really early on in the disease. And after doing all of this work with high glucose, which is one of the metabolic insults that lead to DR progression, I wanted to switch gears a little bit, and that led me to the work of Dr. Julia Busik, and she really is the pioneer in lipid and cholesterol dysregulation in diabetes. So why study about diabetic retinopathy? This disease is actually the diabesity epidemic, that is obesity and type 2 diabetes, is likely to be the biggest epidemic in human history. And if you can see from this slide, it is estimated that in a year 2040, over 640 million adults are going to be living with this disease. And what's even scarier is that 70 million of these individuals are going to have this vision-threatening diabetic retinopathy. So trying to figure out mechanisms that cause this disease progression are really, really important in trying to figure out ways to treat the earlier stages of the disease to prevent the 70 million patients to lose their vision. Now, diabetic pathogenesis, it is, like I mentioned before, the leading cause of blindness among working age adults, and it affects the blood vessels in the retina. This disease really affects all of retina healthy function. My work really focuses on the blood vessels, and that is because the damage that we see to the blood vessels are the ones that we tend to see in the patients that have this vision-threatening stage of the disease. So we believe that it's really important, trying to fix these abnormalities in the blood vessels are really important to stopping the progression of DR. And what I'm showing here is a normal retina to the left, and you can see the fovea, the macula, all are necessary for this high acuity vision. And on the right, you have the diabetic retinopathy retina, and you can see that unlike the nice healthy retina, there's a lot of spots where you have hemorrhages or the abnormal growth of blood vessels, as well as the formation of aneurysms. So all of these pathologies, if they're left untreated, can then cause vision loss in this high percent of the population. So decades of research on diabetic retinopathy pathogenesis has led to a lot of really effective and commonly used treatment options for DR. The first is retinal photocoagulation, and this is laser therapy. So you take a laser beam and you burn small laser spots in the retina, and this really diminishes the demand of oxygen the retina is consuming, so this leads to a dramatic decrease in the amount of abnormal blood vessel growth, which, as I showed in the previous slide, was one of the complications that you see in the end stage of the disease. Since laser therapy, there has been more efficacious therapies, and that is primarily done with anti-vascular endothelial growth factor or anti-VEGF injections. And since 2004, we have different flavors of anti-VEGF injections. All of these are really effective in treating the disease, but they are not perfect. There's still a large percentage of the population with this disease that are not responsive to VEGF. And more studies are coming out that in some parts of the retina, VEGF is actually needed for healthy retinal function. So if you're getting rid of all the VEGF, that could then cause other effects. So my work really right now is focusing on dyslipidemia. And a lot of work has been done trying to understand the effect that high sugar has on DR progression. And not a lot is known about dyslipidemia. So this lipid dysregulation results as an imbalance from regulation of lipid uptake, metabolism, release, and also clearance from the circulation. And this problem is seen in both type 1 and type 2 patients. So in type 2, you have this insulin resistance, but in type 1, you have low portal insulin. So all of these dysregulation in insulin can have really significant effects on plasma lipid levels, lipid profiles, and fatty acid composition. So several clinical trials, here I am highlighting one work, but there's been many clinical trials that have shown that diabetic dyslipidemia is characterized by elevated levels of most all lipid classes. You have triglycerides, sphingolipids, cholesterol. For the purpose of my work, I'm going to be focusing on the role that cholesterol has on DR progression. So very briefly, an overview of cholesterol. This is a lipid composed of a 4-ring system. It is mainly associated with membranes, and it forms these multi-molecular complexes, and they are transported through the systemic circulation, as you can see highlighted here. So the retina is very unique. Unlike other peripheral tissues like the brain, for instance, that primarily locally synthesize cholesterol, the retina actually has 3 different ways to regulate cholesterol homeostasis. So it either does it by systemic control, but it can also produce cholesterol, and it can also eliminate cholesterol. And under healthy conditions, most of the cholesterol has been shown to come in through this retinal pigment epithelium layer. However, in diabetic conditions, you also have cholesterol coming in through the endothelial cell layer. And this is important because in diabetic retinopathy, you have this increase in the cholesterol accumulation, suggesting that we have some kind of imbalance in the amount of cholesterol coming in and the ability of the retina to take it out. So all of these different mechanisms should really emphasize the importance that you have in maintaining endogenous cholesterol biosynthesis. And this is really important because studies have previously gotten rid of all of the retinal cholesterol, and if you do that, you actually have retinal degeneration. So we don't want to promote all of the elimination of cholesterol. We just want to go back to those healthy levels. And one of the ways that you can achieve this is by modulating this key regulator of cholesterol homeostasis known as LXR. And this is one of my favorite nuclear receptors that I'll be talking about today. So there's two isoforms of LXR. You have alpha and beta. These are nuclear sensors, so the way that it works is cellular oxysterols, and these oxysterols are just metabolites of cholesterol. They accumulate as you have more cholesterol, and once the oxysterols bind to LXR, it becomes activated. And activation of LXR then promotes the activation of the reverse cholesterol transport. So it basically activates pathways that are going to get rid of that excess cholesterol in the tissue. And we've previously shown, our collaborators have shown that LXR is an important regulator. So if you activate LXR in STZ mice, STZ is a type 1 model of diabetes in animals. If you activate LXR, you can prevent DR pathology. And what we're showing here is an acellular capillary. This is a hallmark of DR progression in animals. And if you look at panel D, you can see that there is less the white arrows are highlighting the acellular capillary formation, which is increase in diabetes. That's panel C. So you can see that the animals treated with an LXR activator have less of these, suggesting that these animals have less diabetic-induced damage than the diabetic counterparts. So shown here again, all three, both alpha and the beta, as well as the double knockout, are all important, so suggesting that both isoforms are very important in regulating cholesterol. And what we've shown, my data has shown that not only does diabetes decrease LXR in mice, but you can also see that in human endothelial cells. And the way that we do this is that we take human retina, so you have a non-diabetic retina and a diabetic retina. And you can see again the formation of those hemorrhages in the diabetic retina. And you take these retinas and you isolate retinal endothelial cells from them, and with PCR, you then look at expression levels of LXR, and you can see that in the diabetic, there is a decrease in LXR alpha and LXR beta. So looking at these oxysterols, like I mentioned earlier, these are endogenous activators for LXR. So shown here, I'm just showing the ones in green. These are the flavors of these oxysterols that are activators for LXR. So not only is diabetes downregulating LXR, but it's also downregulating, if you look at the oxysterol form here, it's downregulating the activators, the endogenous activators for LXR, suggesting that and this is using a type 2 diabetic rat model, suggesting that diabetes is really having this negative effect in pathways that are promoting the export of cholesterol out of the retina, suggesting why you perhaps have this high accumulation. So the novelty of my work in the Busik lab really stems from identifying this novel signaling pathway, and that's what I'm going to be showing evidence of today. So the one player that we found that activates LXR is SIRT1, and this has been shown previously to activate LXR in non-retinal systems, but the work in the lab that I've been doing really has shown how this player really activates LXR in the retina, and activation of the signaling complex, which I hope to convince you by the end of this presentation, really is a potential therapy to stop the progression of diabetic retinopathy. So a little bit of SIRT1. This is a really interesting enzyme. It is a member of the sirtuin family, so it is NADA plus dependent, and it's really involved in a slew of things, as you can see in this slide. Most relevant to my work, I'm looking at the way that it regulates metabolic syndromes, aging, and of course diabetes. So we have shown with the same method that I previously showed with LXR that if you look at SIRT1 expression in the diabetic retina, it significantly decreased in the diabetic condition. So the hypothesis moving forward is that you have, the way that the mechanism works is that you have activators such as the oxysterols or pharmacological activators of LXR. They bind to LXR, and then this can cause the activation of the reverse cholesterol transport genes, which then can eliminate cholesterol. But you can also repress inflammatory genes. And this is, I just want to spend a couple of seconds on here because this is really important. This pathway is not only promoting cholesterol efflux from the retina, but it's also repressing inflammation. And low-grade chronic inflammation is a hallmark of diabetic retinopathy progression. So activating this pathway is really exciting because you can kind of do two things at once. You can get rid of the excess cholesterol, but you can also prevent inflammation. So the idea is that diabetic retinopathy decreases the amount of LXR activation, which then leads to dyslipidemia by dysregulation of reverse cholesterol transport, and it also leads to inflammation since you have an increase in all of these markers, which I've highlighted here. And of course, that would lead to DR progression. So the bulk of my work looks at endothelial cells, as I previously mentioned. I do this by doing a cell culture model. These are primary retinal endothelial cells. And the protocol that I follow is this paper that I've highlighted here in case anyone is interested. And basically, we are generous to have a meat lab on campus. And they are generous with their samples, and I am able to acquire bovine eyes from them. I then dissect the retina, as I'm shown here, and then these are just the retinal layers, so I'm focusing on the endothelial cells. So we then culture them. The whole process takes about one and a half weeks, and after that process, you have these beautiful endothelial cells, which are relatively easy to acquire. They're economical, and the most important part is that they mimic what we're seeing in the human culture. And of course, towards the end of the studies, we always do human cultures to verify our effects that we're seeing. We verify that these are endothelial cells via two methods. The first is looking at LDL uptake. So this is just a positive control. If you hit endothelial cells with low-density lipoprotein, they will take it up. And that is shown here with the images as well as the quantification. And we also do von Willebrand factor staining. Again, this is published showing that endothelial cells express this protein. So you can see here at different levels of confluency, they're all expressing VWF. So using this model system, I've really been able to dissect the way that LXR and SIRT1 are activating reverse cholesterol transport and inflammation, focusing on RCT. So here, the top just shows the schematic of what we believe is happening. So if you hit these cells with a diabetic relevant stimuli, and that is what TNF-alpha is. So one of the challenges of culture systems is trying to find the right stimulus to mimic what's happening in a very complex in vivo system. So here, TNF-alpha is known to be upregulated in humans. So we are using this model to mimic the inflammation that we see in the human eye. So if you hit the cells with TNF-alpha, you see that there's a decrease in LXR signaling. There's also a decrease in ABCA1, which is part of this reverse cholesterol transport, as well as ABCG1. So interestingly, if you activate LXR with DMHCA, this is a selective agonist for LXR, so it is just like the oxysterols are an endogenous activator, this is just a drug that you can treat the cells with, and then it selectively activates LXR. So you can see, again, that you have this decrease in A1 and G1 with TNF-alpha, but if you treat the cells with DMHCA, even in the presence of TNF-alpha, you can have an upregulation of A1 and G1, suggesting that if you were to treat an animal or a patient that has this high levels of TNF-alpha or inflammation, you can maybe activate cholesterol reverse genes and then promote the efflux of cholesterol from the retina. Looking at inflammation, the other wing of the pathway, again, you have this massive increase with IL-1 beta in both. So the left is DMHCA. This is a selective LXR activator, and you have an increase in IL-1 beta. But then if you treat them with either the DMHCA or the SIRT1. Now, SIRT1 is a selective SIRT1 activator. So here we're looking at the role that SIRT1 is playing. So you can either you can treat them with the LXR activator on the left or the SIRT1 activator on the right. And you can see that in both instances, you can prevent this increase in inflammation that you're seeing because of TNF-alpha treatment. Now, what's really exciting, we see all of these phenotypes in the cells, but if you move on to the animal model, this is a type 2 DB mouse model, you see again the increase in acellular formation. These are the diabetic untreated animals, but if you treat the animals with GW3965, another LXR activator, and we've actually done this too with DMHCA and see similar results, you have a decrease in this acellular capillary formation, and that is quantified in this graph, suggesting that all of the effects that we're seeing in vitro in preventing cholesterol efflux, or promoting cholesterol efflux and preventing inflammation, could somehow be preventing the disease from advancing in the animal. So after this work, we really became interested in trying to find non-pharmacological or different ways of activating the pathway. DMHCA and GW are really great activators of LXR, but there are some adverse effects, and those include lipogenesis. So there's a lot, since you're activating so much removal of cholesterol, that could have negative effects. So I was really interested in the role that SIRT1 is having as a calorie sensing, a nutrient sensing enzyme. So SIRT1 is activated in calorie restriction environments, and recently there has really been a plethora of results and interesting studies showing, especially this recent calorie study, showing the beneficial effects that fasting has on aging and lifespan and prevention of several metabolic induced diseases. So this really, I was really interested by this, because if SIRT1 is activated in these calorie restriction, nutrient deprivation states, could we then use this model to activate SIRT1 and then ultimately activate SIRT1, and then this could give similar effects that we see in the animals when we're activating LXR with pharmacological means. So this led to the hypothesis that perhaps serum starvation could lead to the upregulation of SIRT1 LXR signaling and thus prevent DR progression. So to test that hypothesis, I again went back to the bovine endothelial culture model, and I treated cells with serum containing 0% FBS. And you can see here looking at PCR, so this is expression level, so you have an increase in SIRT1. You also have an increase in A1 and G1, and these two are the proteins involved in the reverse cholesterol transport. And these are always used in my studies as a measure of LXR activity. So we're suggesting that LXR activity is up, and also what's really cool is that you also have a decrease in VCAM1, and this is a measure for inflammation. So here we're showing that if you simply remove serum from these cells for 24 hours, you can have an increase in SIRT1, LXR activity, and a decrease in inflammation. And you also, here I'm showing evidence looking at SIRT activity. So on the left you have expression levels, but if you also look at how much histone deacetylase activity you have in these cells after serum starvation, which is the light gray bar, you have an increase. And the TNF-alpha is a positive control, since we know that inflammation decreases HDAC activity, and that's what you're seeing here. Moving on, trying to really figure out if SIRT1 is mediating these effects, what I did was with siRNA, I got rid of SIRT1, and here I'm looking at cholesterol export. So if you remove SIRT1 with siRNA from this culture, you lose this upregulation of A1 and G1 that you see as a result of serum, suggesting that SIRT1 really is playing this crucial role in modulating the serum starvation effects. So in addition to modulating LXR activity with using A1 and G1, we're also really interested in this because it's also increasing LXR protein levels. And this is suggesting that perhaps it's not only increasing activity, but it's also increasing the amount of available LXR that is around for SIRT1 to activate it. And we are currently working on publishing these results in an upcoming study. So to summarize this portion, we show that fasting and serum starvation can increase SIRT1 and increase LXR. So once you have increase in SIRT1, you have an increase in LXR activity, and then that can lead to decreases in inflammation and cholesterol efflux. So suggesting that SIRT1 is mediating these serum starvation results. And what's really interesting about this is that when you look at all of the fasting studies, most of them, there are mechanisms that are being suggested to regulate these effects, but no one really knows how these beneficial effects that we're seeing due to fasting are happening. So this work is exciting because not only are we are suggesting a non-pharmacological way to activate these beneficial effects that you see with LXR activation, but it's also giving a mechanism of action or sort of an explanation to what is happening with these beneficial fasting-induced effects that we see in humans and non-human primates and cells and a slew of different organisms. And then this is a little teaser for my paper that's coming up, and we've replicated this study in mice. These were not diabetic mice, these are just control mice, and what we see is that there are trends towards increased SIRT1 activity in mice if you just simply fast them for 2 weeks. So a very short fast can have really beneficial effects. And we also see this with our collaborator, and that work is published. They starved their diabetic animals for longer periods of time. I believe it was 6 months. And those animals actually, the fasted animals, no longer had diabetic retinopathy. So they did not, it was not a SIRT1 story, but that just shows evidence that this fasting protocols are really beneficial in preventing DR and the other diabetic retinopathy pathologies. Yeah, so in summary, I just want to highlight one more time the beneficial effects of LXR activation, whether it is through DMHCA or SIRT1720 or oxysterols or the serum starvation data. So the idea is that if you activate the pathway, you're going to activate the reverse cholesterol transport, which is going to regulate lipid metabolism, and you're also going to repress these inflammatory genes, which is going to get rid of that pro-inflammatory low-grade inflammation that you see in diabetic retinopathy. And with that, I would like to thank everyone who helped me with this work, specifically everyone in the Busik lab, Todd Leidick, who helped with all of the retinal cholesterol measurements in the animals. The Grant Lab, they are the ones that are really focused on the fasting side of it, and that's the paper that I referenced earlier. They have done a great job characterizing intermittent fasting and diabetic retinopathy progression, as well as our funding sources. And I would love to hear from you. If you have any questions, please contact me. I have my email and my Twitter handle here. With that, I will be very happy to take any questions. Thank you so much.
Thank you, Dr. Hammer, for your informative presentation. Yes, now it is time to start our live Q&A portion of our webinar. To our audience, if you have a question you would like to ask, please do so now. Just click on that Ask a Question box located on the upper left of your screen, and we will answer as many of your questions as we have time for. So we have some great questions coming in. Dr. Hammer, let's start with this one. Does serum starvation modulate LXR activity that is dependent on SIRT1?
Great, thanks, Christy. So that is a good question. I don't want to say that it's completely dependent on SIRT1, but I do have some work showing that if you get rid of SIRT1 with siRNA, you lose the beneficial effects that you see with A1 and G1 upregulation as a result of serum starvation. So although it might not be completely dependent, we do have some evidence showing that it is playing, that signaling pathway is playing a significant role in this.
Can you let us know if serum starvation can affect cell death?
Right, yeah, so that is kind of the question that I get asked a lot. So if you are removing serum, you would maybe think that you could be affecting cell death. And we've looked at this in our retinal endothelial cells, and what we see is that there's no change in cell death after 24 hours. However, if you leave them in this serum starvation media for longer than, I think I've gone up to 96 hours, then you do start to see cell death at that extended period of time. But at our time points, which are 24 hours, we do not see any cell death. And we've also looked at other health functions such as mitochondrial respiration, and there is no difference in that measure either.
Thank you, Dr. Hammer. We have a question coming in. What is the role of RXR in SIRT1?
So that's an interesting question. So I'm not too familiar with the role of SIRT1 and RXR. I do know that LXRs form a heterodimer with the retinoid X receptor, which is the RXR, and these are in the literature known as an obligate partner. So this has to be this heterodimer has to be formed in order for LXR to transcribe ABCA1 and G1 and other targets. I'm not sure, however, how that interaction is playing with SIRT1. It would be interesting to see if once SIRT1 deacylates LXR, if that interaction with the RXR is no longer needed, or is it altered in any way? But that is an interesting question to further investigate.
Does systemic cholesterol affect retinal cholesterol levels?
The reason why I paused for that, so the layer of the choriocapillaris facing the RPE has openings that enable the delivery of nutrients from the systemic circulation. So we do know that the systemic circulation, the available of cholesterol in the systemic circulation could affect the RPE intake of cholesterol, but we've also seen clinical trials where systemic control of cholesterol has not been beneficial in helping patients that have diabetic retinopathy, and that is why we think that the way to modulate the cholesterol levels is to really locally do this. So we want to directly target the cholesterol levels in the retina as opposed to globally treating the cholesterol increases, since we do have evidence from human clinical trials that have shown that if you do that, if you go that route, you don't really have an effect to the cholesterol levels in the retina. So we really want to, when we're talking about cholesterol in the retina, we really want to focus to the tissue specific rather than going the systemic route.
Thank you, Dr. Hammer. Now, when you talk about fasting, for how many hours a day?
So when we talk about our cells, so if you're just looking at a petri dish, and I did my studies for 24 hours. If you are talking about animals, we have fasting studies that range from, I've seen from 2 weeks all the way to extended periods like 6 months. If you're talking about humans, that is a completely different system. So there are many different regimens out there, and we are not really suggesting that this is something that should be done in the human system. We are just providing some evidence for the beneficial effects of modulating cholesterol. So there is really a wide range of steps that need to happen before we can really suggest a fasting regimen or something that would work in humans. But in the animals, there's really a really wide difference in studies that have shown beneficial effects. And I really think it's going to come down to the individual person rather than just having one protocol that really works for everyone.
Now, while we're on the topic of fasting, how is caloric restriction different from intermittent fasting?
So calorie restriction and intermittent fasting, these have been kind of a hot topic for a while in in research and in medicine. And calorie restriction, that's one of this research dates back many, many years. And when you look at the animals, or like I mentioned in the talk, looking from cells all the way to non-human primates, if you do calorie restriction studies, you can prevent aging, you can prevent a wide range of metabolic associated syndromes. However, it is really hard to implement in humans since there are negative effects that have been associated with calorie restriction, such as malnutrition, problems when it comes to fertility, things like that. So intermittent fasting seems to provide a more approachable way of implementing the positive effect that you have from calorie restriction. So intermittent fasting simply means that you are modulating the amount of nutrition to a specific time. And sometimes, I would say most of the time, that does come with some sort of less calorie intake, but not necessarily. And the benefits of the intermittent fasting is that you have the periods where you are normally consuming meals, followed by a period where you're not. So you have that active recovery stage, and you allow the your metabolism to kind of recover from that or that no nutrient intake when you are feeding, as opposed to calorie restriction, which is a more dramatic way to cut down how much nutrient you're consuming. And I know, for instance, the National Institute of Aging has a really nice section on their website on the benefits of calorie restriction and intermittent fasting, and they also have all of the studies that have been done to really back those claims up. So it's a wonderful resource for someone who has more questions about that topic.
Great. Dr. Hammer, could you repeat that website again, or where we could find that information?
Yes, it's the National Institute of Aging. It's through the NIH, and you can just do Aging Institute at the NIH, and it should come up.
Excellent. Thanks so much. Now, our next question. There are animal and human studies that suggest SIRT1 is activated by invoking intermittent fasting, carbohydrate restriction, ketosis, and or exercise. Do you have any idea how the other protocols affect LXR?
Yeah, so since LXR has two arms of its pathway, I could also imagine LXR having this beneficial overall effect by modulating the low-grade chronic inflammation. So that is kind of the one pathway that my mind is going to. I'm sure there's other mechanisms that it could be having its positive effects, but really by not only promoting the cholesterol efflux, but also getting rid of this low-grade chronic inflammation could be another mechanism of action that, with SIRT1 or not with SIRT1, independent of SIRT1, could be having these beneficial effects. And I hope that answered the question.
Thank you. Have you looked at other markers aside from IL-1B to measure inflammation?
Yes, we've looked at IL-6, we've looked at VCAM and ICAM, and we see similar effects. With the inflammation, we want to, the way that we have seen before is that it doesn't have to be, but we have looked at all the ones that are NF-kappa B dependent, and that is the one that we've seen previously published that LXRs having a direct effect. But yeah, so we've looked at IL-6, VCAM, ICAM, and we've also looked at MCP1, and we have, all of these are also highlighted in the Hammer 2017 eBiomedicine manuscript, and we have more markers shown there.
Okay, let's go to this question. Let's see. Can you explain to us how LXR and RXR work?
Yeah, so I don't know too much about RXR. I do know that it does, in order for LXR to activate A1 and G1, for instance, those its downstream targets, it has to form a heterodimer with RXR. Now, I don't know if this interaction is necessary once SIRT1 deacylates LXR, or if once SIRT1 deacylates R, is it necessary to form this dimer? I'm not sure. It would be interesting to definitely look at that mechanism for sure.
Now, you mentioned adverse effects to LXR activators. Can you explain what these are?
Right. So one of them, the main one, is that it provides activation of RSBP1, and this can lead to lipogenesis. So you don't want that occurring. That could be very bad, and that's actually why some of the studies looking at LXR activators in humans have not gone very well because of that negative effect on the sterol regulatory element binding proteins. And yeah, so that's kind of the main one.
Thank you, Dr. Hammer. Now, do you have any final comments for our audience?
So I would mostly just want to thank everyone for registering and for listening to my presentation. And if you would like, if you have any other questions or would like to ask me anything, my email is shown here and also my Twitter handle. And I would love to hear from of you. And just wanted to give one last shout out to my PI for the support, not only in the lab, but also supporting me with this webinar. And it's been really nice to have that support from the lab. So I just want to thank that. And Gipco and Christy for this opportunity. It's been a lot of fun, and I really appreciate it.
And we would like to thank you again, Dr. Hammer, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. And before we go, we would like to do one more thank you to our audience for joining us today and for their interesting questions. Now, we have a few questions we weren't able to answer today, and any of those questions submitted during the on-demand period will be addressed by Dr. Hammer via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. You will now be redirected to the registration page of our next webinar titled An Automated Cancer Organ Culture Platform to Accelerate Research and Drug Screening, presented by Brooke Schuster on January 30th at 9 a.m. Pacific Time, 12 p.m. Eastern Time. We hope to see you there. That's all for now, and have a great day.